Just to quickly introduce Robbie, he's a postdoctoral researcher at the Department of Methodology and Statistics at Tilburg University and a member of the Meta Research Center. And he's also a BITSS catalyst, and we've worked with him for quite a few years. So it's nice to see you again, and take it away.
Thank you for the introduction, and welcome everyone. So in this presentation, I will tell you something about a recent paper that I wrote about how to analyze data of a multi-lab replication project. So this presentation is in some sort of tutorial kind of way set up. So first, an overview of my presentation. I will start with describing what multi-lab replication projects actually are. Then I will say you something about how they are usually analyzed in practice. And I will present another method for analyzing these data. And this is individual participant data meta-analysis. Then there are two different types of this IPD meta-analysis, a one-step approach and a two-step approach. So that's what I will also introduce, and then apply this IPD meta-analysis to an example, a registered replication report by McCarthy and colleagues. And I will end with a discussion. So first about these multi-lab replication projects. So the idea of these projects is that some prominent effect is selected, and this effect is replicated in multiple labs across the world. And then there are usually two aims. The first aim is that researchers want to study the replicability. So replicability means, can the effect be replicated? And the second aim is whether an effect is robust. So does the effect depend on contextual factors? So for example, it can be the case that an effect does replicate in the US, but doesn't replicate in Europe. And then this can be an interesting finding that we can also use later on for setting up new studies. So examples of these multi-lab replication projects are the registered replication reports, so RRRs. So in these RRRs, usually one effect is replicated across multiple labs. And there are also these many labs projects that are usually initiated by the Center for Open Science, where multiple effects are replicated within a project. So currently, if you look at the two major outlets for these multi-lab replication projects, which are Perspectives on Psychological Science and AMPPS, then there are 12 RRRs published. And in the different many labs projects, there are in total 60 effects studied. So 60 effects replicated. So how do these data look like? And how are these analyzed? How are these data usually analyzed in these multi-lab replication projects? So what we have is we have participant data, of course, and then we also have, we know that these data, these participant data are nested within the labs. So these participants are, of course, these data is collected within a lab. And then what is usually done is that based on this participant data for each lab, some summary statistics are computed. So for instance, a mean or a mean difference. And these are then meta-analyzed in the meta-analysis. And this is also the way how you would usually do it in a meta-analysis, so not in a multi-lab replication study, because then of course you do not have information about the participant data. You only have the summary data, and this is the data that you can use in your meta-analysis. But in the multi-lab replication projects, you also have the participant data because you collected the data yourself. And therefore, this offers an opportunity to directly analyze all the data of all the labs together by using the participant data rather than the summary data. So if we look at these multi-lab replication projects that I showed on the previous slide, 72 in total, then 97.2% of these multi-lab replication projects used summary data. So didn't use the participant data, which is suboptimal if you only use the summary data whenever the participant data is available. So IPD meta-analysis. So what it in principle is it is applying multi-level models to the data of the participants, and the data of these participants is nested within the studies or within the labs in a multi-lab replication project. And advantages of this IPD meta-analysis over conventional meta-analysis as often used is that, first of all, statistical power is generally higher to find a treatment effect. And that's because if you use the data of the participants, you're basically just using more data than if you use the summary data, and therefore you have higher power. Another advantage is that if the same data is used in multiple studies, then you can also figure this out if you do an IPD meta-analysis. Something else that you can do is you can make sure that the data analysis is standardized. So in each and every study or in each and every lab, you can handle missing data in a consistent way, and you can also make sure that the same procedure is used for removing the outliers. There's also no need to transform effect sizes. So what you generally often see in a meta-analysis is that you have different effect sizes from each study, and you need to make sure that these all are on the same effect size measure. But if you have the individual participant data, this is not needed because you can simply compute the effect sizes yourself. And finally, and that's I think the most important advantage of IPD meta-analysis over conventional meta-analysis, is that you can also use participant level moderators to explain the heterogeneity. So in the meta-analysis, we generally, we can only look at moderators that are measured at the study level or at the lab level. And in IPD meta-analysis, we can also use information of the participants themselves. So we can use information about, for instance, the age of the participants, and we don't have to use the mean age as we need to do in conventional meta-analysis. So then there are two approaches for the IPD meta-analysis, and both approaches allow drawing conclusions at the participant level. So in the two-step approach, then effect sizes are first computed per lab, and then these are synthesized using the conventional meta-analysis methods researchers are usually already familiar with. That's also an advantage of this approach. A disadvantage of this two-step approach is that we are still, in the meta-analysis, we are still using summary data. We are not really analyzing this individual participant data. We only do this in the first step. So therefore, we still have this problem of low statistical power. Then there is the one-step approach, and the one-step approach, the participant data is modeled directly using a multi-level model. And advantages of this approach are that it's a very flexible model. So you can add a lot of predictors, for instance, and it has larger statistical power. A disadvantage, on the other hand, is that the model is more complex, and this can, for instance, result in convergence problems that can be hard to solve. So that's a bit tricky about these one-step IPD meta-analysis models. So I will illustrate these models using an example, a registered replication report by McCarthy and colleagues who replicated a study by Skrill and Wire on assimilative priming. And assimilative priming refers to the idea that exposure to priming stimuli causes subsequent judgments to incorporate more of the qualities of the primed construct. So this sounds, at least to me, quite complicated, but I hope that this becomes clear if I explain the procedure of the replicated experiment. So what is done in this experiment is that participants are first asked to perform a sentence construction task, and these sentences contain either 20% or 80%. In 20% or 80%, they describe hostile behavior. So the control condition is the 20% sentences with hostile behavior, and the experimental condition is the 80% condition. And then after this sentence construction task, participants are asked to read a vignette about a person who behaved in a hostile way, and then they had to rate his behavior, whether it was perceived as being hostile. And then the hypothesis is that participants who are exposed to a larger number of sentences describing hostile behavior, so the 80% condition compared to the 20% condition, whether they would rate the person's behavior as more hostile. So we have two different groups. So we can use a raw mean difference as effect size measure, where a positive difference indicates that the hostility rating was larger in the 80% condition. So in this RRR, 22 labs participating, and this yielded a sample size of a bit more than 7,000 participants. So let's first start with the two-step approach, how this works. So in this two-step approach, what we're doing in the first step is we are fitting a linear regression model. So just a standard regression model, where in this case, the Yj refers to the score on the dependent variable of participant J. Then the phi over here is a fixed lab effect. And then this theta xj refers to the treatment effect, where xj j is a dummy variable, where 0 refers to the 20% condition, and a 1 refers to the 80% condition. And then the epsilon j is the sampling error. So what needs to be done is this linear regression model needs to be fitted to each lab. And we can do this in R with this function, with the LM function. And we do this for each lab. And if we have done this, then in the second step, then we extract the estimated treatment effect. So the theta hat, we extract this from the output of R. And then we meta-analyze these with a conventional meta-analysis model. So this can, for instance, be done with the metaphor package using the RMA function, where you use as effect size these estimates of the theta hat, so the estimates of the treatment effect, and you also need to use the need to know the variances, and the variances are also available in the output of the LM function. So this is how it works for the two-step IPD meta-analysis. Then for the one-step IPD meta-analysis, we now fit a single multi-level model to the data. So it looks very similar as what we saw before, but now we have the Yij. So I now refers to the lab. And we also again have a lab effect. We have a treatment effect and the sampling error. And then what is important over here is that there is one very controversial decision. And that is whether you should treat this lab effect as a fixed or as a random parameter. So if you would treat it as a fixed parameter, then this means that each lab, that for each lab we estimate a separate effect. So this results in a large number of parameters in the model, especially if there are a large number of labs. Then if you say, okay, we go for random effects for the labs, then you assume that these labs are sampled from a normal distribution. And this can also be a tricky assumption because we never know for sure whether these lab effects are indeed sampled from a normal distribution. It could also be another distribution, and therefore this is a quite controversial decision. So what I will do, I will show you the results for the random lab effects, because it has another advantage, and that's that we can generalize the results to the population of effects. And if you want to do this in R, then for instance, you can use the LME4 package using the LMER function. And then if you use this LMER function, the first part is the same as we saw before. And then this part between brackets refers to the random effects. So what we are doing over here is the lab specifies that we want to have a random intercept. So it means that this phi I, the lab effects, can differ from study to study. And this x over here refers to that we want to have a random slope. So it means that the treatment effect can differ from study to study. And this is what you call the heterogeneity in the meta-analysis. So the results, if we apply these methods, so first of all, the random effects meta-analysis, that's what you're seeing over here. And these results exactly coincide with the results that are reported in the paper by McCarthy and colleagues. So this is very good news that I was at least able to reproduce these results. And then what you see if you compare the results of the random effects meta-analysis with the other two IPD meta-analysis models, then you see that the results are very similar. One thing that's remarkable is that the confidence interval for the one-step approach, that this confidence interval is a bit smaller, and that's also what you would expect, because for the one-step approach, we are using all information that's available. We are using the information of the participants and not the summary data. So something else that you might want to do, because that's usually an aim in meta-analysis, is that you want to include moderator variables. And if you include these moderator variables, then you fit a slightly more complicated regression model. And in this regression model, what you do is you include the main effect of the moderator W in this case, and you include also additionally the interaction effect between the moderator W and the treatment effect X. So how do you do this in R? Again, you can use the LM function and now include the H. So H is our moderator in this case, so the main effect of H is included, as well as the interaction effect over here. So then based on this output, we can do it in a similar way as we did before, but now we are meta-analyzing the interaction effects between the treatment and the moderator. So now we are meta-analyzing these gammas that were estimated in R, in this LM code. So again, we do this with the metaphor package, and then we would get the results for the two-step IPD meta-analysis. Then you can also do this using a one-step IPD meta-analysis with a moderator, but there it becomes a bit more complicated, and that's because we can make use of an additional feature, and that's that we can disentangle the within and between lab interaction. So we can see what is the interaction effect within a lab and what is the interaction effect between labs. And in order to do this, we need to use something that's quite popular in multi-level modeling, and that's that we need to use group mean centering. And group mean centering implies that we are subtracting the mean score on the moderator variable per lab. So we compute per lab, we compute the mean score of the moderator, and then we subtract this from the scores on the moderator. So if we look at this regression equation, it's more complicated than we saw before, but the new elements that were added are the main effect of the moderator variable. We have the interaction effect between the treatment variable and the group mean centered variable. So the difference between the scores on the moderator W and the lab mean. So this provides us information about the within lab interaction. And then for the between lab interaction, we include the interaction effect between the lab mean, the MI, and the treatment variable, the X, Y, J. So how do you do this in R? Again, we can use the LME4 package. We add a couple of additional terms. We add our moderator variable H. We add an interaction effect between H minus the GM means the group means, so the lab means in this case. So this is the interaction effect between the treatment X and the group mean centered variable. And we can also include the interaction effect between the treatment variable and the lab means. So if we look at the results, if we apply these different methods, now first of all, what we see is that for the random effects meta regression approach, the ones in the line in red, then you see that there is no effect of mean age in this case. So we can see this because 0 is included in this confidence interval. If we look at the green lines refer to the interaction effect within a lab. Then we see that for the two-step and the one-step approach, results are very similar in this case. And we see that the interaction effect within the labs within the labs that this interaction effect is statistically significant because the 0 is not included in the confidence interval for both in the two-step approach and also in the one-step approach. Then if you look at the interaction effect between the labs, then we see that H does not have, there is no interaction effect between the treatment and H, because here again, the 0 is included in the confidence interval. So to conclude, usually conventional meta-analysis models are applied to analyze data of multi-lab replication projects, but this is suboptimal because we do not make optimal use of the available data. We do not use the participant data that are available. A better approach is especially to use this one-step IPD meta-analysis approach, because there we make use of the participant data. But there's also a drawback here, and that's that you may encounter convergence issues that might be difficult to solve. So if you encounter convergence issues, then possible solutions are, first of all, to simplify the model, if that's possible. And if that's not possible, then as a last resort, you could say, okay, I go for a two-step IPD meta-analysis approach. So some possible extensions for this IPD meta-analysis. So it can, for instance, be used in internal meta-analyses that are quite popular within psychology. So in psychology, there are usually multiple studies included in a paper. And then researchers sometimes want to meta-analyze all these studies in their paper. And they nowadays usually use the conventional meta-analysis models for this. But they could also use IPD meta-analysis models because they have the data, the participant data, because they collected these data themselves. Other extensions and advantages of one-step meta-analysis, yeah, it's very flexible. So you could also say, I would like to add an extra random effect, for instance, and you might want to add an extra random effect if you want to take into account that, for instance, some studies are conducted in Europe, whereas other studies are conducted in the US. That can be done with an extra random effect, which is more difficult to do so in conventional meta-analysis. And finally, that's what I hope, is that sharing this participant data becomes the norm, such that in a couple of years' time, we don't need the conventional meta-analysis models anymore, but we can basically rely on this IPD participant data meta-analysis. Thank you for your attention.
All right, thank you, Robbie. Okay, we've got about 7 minutes or so for questions. I see one in the chat from Michael. Would you like me to read that, or do you want to go ahead and read it?
Let's see.
Can you see it?
I can see it. If I can comment on the methods for combining participant level and summary level data. Yeah, okay, so this is a very good question, because also later on what I say in my on my last slide, if these data become available, the individual participant data, then we also still have in the literature summary data for which this participant data is not available anymore. So there are methods for doing this, for combining these types of data, and they are more popular in medical research, but I haven't seen these being applied in psychology research. And for instance, one approach that I have seen that people take is that they just use the data, the summary data of these studies, and then simulate data given the estimated parameters of these studies. So whether this makes sense or not, yeah, this would really depend on the assumptions that you make in the simulation. So that's I think a very interesting topic for further research.
All right, and if people have other questions, you can just type them in, or we can unmute you. You might also be able to unmute yourselves. I'm actually not sure. So if you want to do that, you can. Throughout your career, have you seen sharing participant data become more common? Like how, can you give like a rough estimate of like how common this is right now? And yeah, just curious.
Yeah, so it's hard to say a percentage, but what I see is that especially I think especially because the open science framework, for instance, has become quite popular among I think especially psychologists, but also other research fields. And I think more and more people are going to share their data, but this is also not without risks because you have to be careful and take into account the privacy of the participants. So it's definitely becoming more popular, but it's still not very common. And I think also there are also very many researchers within psychology, for instance, that don't ever share data, that also don't pre-register a study. So it becomes more popular, but we are definitely not there yet that we can use only IPD meta-analysis.
Sounds about right.
Just a quick questions. I think I'm really interested in what Mike has suggested. Would you be able to send us some recommendations or paper that you have seen that use simulations to combine a summary level data with participants level data? I think this will be very useful.
Yeah, I can do so. I don't have any papers right now in mind. I mean, from the top of my mind, but there are definitely papers about this. So I can take a look. You can also take a look yourself. I mean, I think if you search for combining aggregated data with IPD meta-analysis, I think then you will also be you will get some good hits.
Great. Yeah, so it looks like you can unmute yourself. So if we've got about 2 or 3 more minutes, if you prefer to just unmute yourself and ask a question.
I have another question. I'm happy to yield the floor to others, so give it a sec.
And actually, when asking a question, can you, I think people can tell who is speaking, but please say your name and maybe a couple words about your background, your discipline, or where you're from. But yeah, Michael, go ahead.
Okay, so Mike Frank, Stanford Psychology Meta Research. You didn't find a kind of major kind of failure mode for the summary level meta-analysis here. But it seems to me that there might be some very well, that there might be cases in which you see kind of pretty divergent results between these. So I had one in mind, which was a case, for example, where you had kind of disparate cross-sectional age samples, and then you combine kind of as individually flat regression coefficients, whereas if you had actually done the IPD, you would see an age slope. So kind of simplified example, you have kind of 2-year-olds and adults, and they're in 2 different studies, and neither one shows age-related variance. But if you put them together, of course, there's huge developmental change. So that would be an example of a case where this would fail. But I wondered, because you'd been thinking about this, if you had other failure modes in mind, or cases where you thought that these 2 methods would diverge importantly, and where IPD would be kind of a key strategy to use.
Yeah, very good question. So I also have in this paper, and there's a link to a preprint over here. So in this paper, I have an artificial example. That's an artificial example of a situation where you get completely different conclusions based on an IPD meta-analysis and based on a conventional meta-analysis. And in this situation, that's what you call making an ecological fallacy, because then you are, if you would do this, if you draw conclusions at the meta-analysis level regarding the participants, then you would get to the wrong conclusion. And that's what you are referring to. So I came up with an artificial example. I created an artificial example in the paper, and I think what would also be interesting, and that's also something I would like to work on, is in these many-lab studies where we also have the individual participant data because it's publicly available. they generally also use these conventional meta-analysis methods. So it would be very interesting to apply this IPD meta-analysis to these studies and then to see whether you get different results. So that would definitely also be interesting for future research that I likely plan to do in the future.
Okay, we're actually a little bit ahead of schedule, so we do have a couple more minutes for questions, or we can just move on to the next session. So if anyone has any more, just let us know in the next 30 seconds or so. And if not, there's the Slack channel will be open. And I think Robbie's on the Slack channel, so you can continue the conversation with him there. And then as Ted mentioned earlier today, we'll be opening more breakout rooms tomorrow. One kind of like the one earlier where people are just randomly thrown together, and then one at the end where you can kind of self-select into a themed room. And so if people want to keep talking about this at the end of the day tomorrow too, that's a good place to do it.